Recordings in progress. So welcome. Yeah, several of you have worked with Michelle and myself over the past 3 years now in one way or another to bring more attention to water quality issues on the Lower Raritan. And as part of that, some of you have been out in the field with us doing water quality sampling and data gathering on the Lower Raritan. Tonight, what we're going to be doing is giving a little bit of an overview of some of our findings the last couple of years. We're going to talk about, and get some input, hopefully, from you all about how we can improve our communications that we send out on a weekly basis during the summer months for our water quality monitoring. We'll talk a little bit about how we are making the link between water quality, pathogens, and stormwater. We're also going to talk about how we can improve our data sharing of the data that we gather. And we're going to talk about how you can get involved further in this work. So we have a number of partners who have supported us either with financial donations or otherwise with gifts of time. So Rutgers has been a strong partner with the LRWP. Interstate Environmental Commission has provided the analysis of our samples for weekly monitoring. The Watershed Institute provided a grant for us last year to support our Spanish language outreach and engagement of Spanish-speaking communities along the Raritan. And we also have been working with the New York, New Jersey Harbor Estuary Program. They've provided a small grant as well to support delivery of our samples to the lab in Brooklyn every week. So I also want to mention partnerships with Rutgers Fehrenfeld Lab. Nicole Fehrenfeld and her students have done genetic testing for us of the samples. And we also are cultivating partnerships and relationships with the Environmental Protection Agency and the New Jersey Department of Environmental Protection. That's a new contact that we have this year, and we look forward to working with them to start to zero in on some of the pathogens issues. So what is it that we've been doing the last 3 years going into our 4th year? Well, we've been working with civic science monitors throughout the summer months, the warmer summer months, to first we train them with EPA standards and approaches to water quality monitoring, which then supports them in a quality assured fashion to collect data samples, water quality samples on the Raritan at 6 different locations, starting upstream in Piscataway, going downstream to South Amboy. So what we're interested in is understanding the impacts of a variety of different parameters on our water quality, but specifically with respect to disease-causing bacteria or pathogens. I'm going to open up a quick poll. I have a sense that you all will answer this question the same way. I'm going to ask here whether you were aware of this monitoring. And if you could just take a quick minute, I'll give you maybe 30 seconds or so to answer this.
And while you guys are answering this, I want to make sure to give credit to Brenda, who has all the maps that you're going to be seeing here. Were produced by Brenda Allen. Is that Brenda's last name? Brenda Allen, who's also with Rutgers University with the Center for Remote Sensing and Spatial Analysis. And she She produces these great maps for us, a lot of times on demand, last minute, when we ask them for it.
Yes.
So thank you, Brenda.
Yeah. Thank you. And Brenda is on right now. So can you see the poll results? It looks like
Yeah.
everybody who answered said yes, they are aware of our polling work or of our monitoring work. Okay, great. Moving on. So, how our I have so many screens up here. So how our water quality data is collected. Do you want to talk about that, Michelle?
Sure, yeah. There's a little gray box in the middle there, but I can talk around it. So the way our data is collected is we are going out. We collect our samples from May till September. We have gone into October, and we're going out weekly on Thursdays, every Thursday. We're part of a large effort in the New York City, New Jersey metropolitan area where volunteers all over the region are going out and grabbing water samples from their local waterway so that we can all be telling the public about the health of and safety of our local waterways. So what we're doing is we are collecting pathogen samples. If you see that little arm and that little hand there, we're grabbing water samples. And we are taking those samples at each site, and we are rushing them off to our lab, which is actually in Brooklyn, because there is a 6-hour holding time. We have to be able to get those samples to the lab within 6 hours in order for them to count and for them to do their analysis. And then we are using grab samples, they're called, but then we're also using what's called a YSI, which is a probe that you dip in the water, and that is wonderful because you get instantaneous readings for water temperature, salinity, dissolved oxygen, a whole host of parameters that we can then, collect this data at each one of these sites. We're recording the data right now on paper. We're hoping this year to do that a little bit differently and hopefully be able to have the data digitally and download it. Some of our sites, we are going off of docks, like you see here on the left. We're taking the samples of the docks, but for some of our sites, we are donning waders and we are wading into the water and taking those water samples. So what are we looking for why are we sampling for pathogens? We are the results come back as fecal coliform. We get fecal coliform results, and we get enterococcus results. And essentially, what we're looking to see is how safe are these waters? How safe are these waters for recreation purposes? Fecal coliform is an indicator of pathogens. So is enterococcus. There's total coliforms. There's all sorts of total coliform bacteria. Fecal coliform is specifically derived from feces, animal feces, human feces. But we are more interested in the enterococcus because that is the standard for saline waters. And much of the Lower Raritan River where we are sampling is saline. So what is enterococcus? It's a spherical shaped bacteria found in warm-blooded animals. And it is, as I said, an indicator of human pathogens. Enterococcus within itself is not dangerous to people, but it's an indicator that there may be viruses, other bacteria in the water that will make us sick, gastrointestinal issues, skin infections. So that's why we use enterococcus and EPA. That's the standard that they recommend. It survives in marine waters. When you sample in fresh waters, the indicator is E. coli, but we're mostly in saline waters. And then the standard that we're looking for is the DEP standard for primary contact recreation, which is swimming, fishing, is 104 colony forming units per 100 milliliters. And what you see on the right here are these Petri dishes with these little colonies of bacteria. And those are actually counted in the lab by Jessica Bonamuso, who is our lab manager. So that's for a single sample. Each sample has to be below 104 colony forming units. It's considered below the standard. 104 colony forming units, it's considered above this, not meeting water quality standards. They also look at a 30-day, what's called a 30-day average, or a 30-day geometric mean. 5 samples over those 30 days have to be below, the mean has to be below 35 colony forming units. So that's how the water quality standard is assessed. So did you want to take this one, Heather?
Sure.
Okay.
So every week we evaluate our samples, per, whether they're below or above that 104 colony forming units per 100 milliliters. So we report out both for enterococci levels, which are the standard that we're evaluating, and then we also report out for the amount of for fecal. So as part of our communications, we're interested in sort of a clear statement, without necessarily the science behind it, to let folks know that the waters are more or less safe or much less safe. So we use color-coded frown faces for those dates, those sampling dates and sites that are above the 104 CFU per 100 ml. And then we use the green smiley faces to indicate those samples that have red at below the threshold, the 104 colony forming units. So one of the reasons that we use the smiles and frowns is, in addition to the colors, is for folks who might have color blindness. We also use the smiles and frowns as symbols in the case that we have a variety of different languages spoken throughout the Lower Raritan region. We do communicate out in both English and Spanish, but we want to be able to reach communities that maybe English or Spanish isn't their primary language. So we want them to have a sense for whether or not the sites at which we sample are meeting the suitable enterococci levels for primary contact. So we also provide information about the time sampled and the date. On this particular snapshot image, we put this information out every Friday. So we do the sampling on Thursday. The samples incubate for about 18 hours. The data is then delivered to us Friday afternoon. We basically transpose it into these sort of snapshot report card graphics to put out on Friday afternoons so that folks have a sense for their weekend recreation activities as to whether or not maybe jet skiing or kayaking or fishing is what they want to do, depending on whether they see a smiley face or a frowning face. You'll see a note in the lower left-hand corner here. Occasionally, we have reporting out at TNTC. That's when there are too numerous to count in terms of the colony forming units, in terms of the units. And there are none reflected in this image, but that's what that TNTC means. Also, every Friday, our results have not yet been certified by the EPA. So when we report out, we have to put a qualifying note saying these results are preliminary and awaiting quality control. So we've gone through our own quality control, training and monitoring, as we were doing the data or the sample catching. And there's quality control at the lab, but then there is an extra set of eyes on all of our data and our methods by the EPA, and that's then certified at the end of the year to basically evaluate the whole package of data that we've collected throughout our summer monitoring months. So quality control is not basically predetermined or determined just because we have those training methodologies under our belt. It has to be assessed by EPA after all the data has been reviewed. So the next polling question is, have you seen our report card snapshots in this format? I'm going to open up the poll here. Okay, can folks see the poll?
Yeah.
Yep, great. Okay, there are lots of yeses, a couple of nos, a couple of no answers. Okay, I'm going to close the poll in just a minute here, just a couple of seconds. Okay, I'm going to close it. Okay, if folks cannot now see that they have not seen the results. Okay, so can you see the?
Yeah, we see the results.
So we have 5 folks who say yes, 2 who say no, and a couple, probably the folks who are calling in, who did not answer. Okay, great. So we are super interested in sharing out the pathogens results in the context of conversations with our Raritan adjacent municipalities around stormwater management. So what you see here is sort of a possibly new and improved map for 2022 that situates our smiley and frowny faces in their locality. So you get a sense for where some of the bacterial loading may be coming from. And that may also give a sense for where some of the green infrastructure or stormwater management interventions might be most appropriate at certain times. So we are sharing these, or expect to be sharing these images. This is a map that Brenda prepared for us with our communities as we talk with them about their stormwater management. And talking with community members too about how to understand the stormwater data, I'm sorry, how to understand the work that they're doing to manage stormwater in the context of improving water quality. So what we're hoping to do is take our data, do interventions, do more sampling, and hopefully we'll see improvements over time in the water quality that we're in the water quality monitoring data that we have. So here's
And
another.
I just want to make clear, this is 1 week's snapshot. This is not a summary of our long-term data set.
Yep. And apologies. So I have so many screens up, it's hard for me to see the actual slide right now because I have all the poll questions open. So I'm opening up another poll question here. How have you used the water quality data, if at all? So I have not used this data. The info helps me decide which activities I or my family should engage in on the water. I share the information on social media. I share the information with my town. I tell my friends and family about it. I use the information for school or teaching or other. Okay, I'll just give another couple seconds. Looks like we have a good mix of responses here. Okay, I'm going to close the poll and I'll share it out. Michelle, can you see the poll results?
Yeah.
Okay. So
Our first impact, our first data collection. This is all the preview. You guys are seeing a preview of the public poll that public survey that we're going to ask for your help with.
right. So a few folks have shared with family or friends. A few have used it for teaching. I know for myself that it's all a case of all of the above. We absolutely have taken this information to the municipalities. We've used it in teaching, shared on social media, and it also it has absolutely helped me decide, whether we'll go out on the water on a particular day or not. So, okay. So another quick poll question here. We shared the 2 weekly snapshots here, how we summarize the data. Whoops. And we want to get a sense from you as to whether you prefer our table with green smiles and red frowns, our river map with green smiles and red frowns, if you think both of them are useful, or if you don't like either of them.
This is really important. We're really interested in making sure that we're communicating out this data to the public effectively. So definitely, if you haven't participated so far in the poll, this would be one to participate in.
Yep. And even if you haven't seen this data before, if this is your first time interacting with it, feel free to answer based on just your initial sense here. Okay. I'm going to close the poll. Share out the results. What are our results, Michelle?
Well, it looks like 22% say I believe the map. The second one was the map. I can't see the full
Yep.
question.
Okay, the river map with green smiles and frowns.
Yeah. And then, but 56% say both. That they like both the chart and the map.
Great. Okay. Okay. It's a different kind of storytelling with each, but I do think that, it could be effective to have both out there. Okay. So now we'll get into discussing the data itself, which I think is why most of you joined us tonight. So,
yeah, so we have been sampling for 3 years. Unbelievably, we're going into our 4th year of sampling. So this data presented here is the summary of the last 3 years of data by site. So on the left-hand side, there's a chart that shows each of the sites, each of our 6 sites, and the percent of the time that it exceeds the water quality standard. And again, that water quality standard is 104 colony forming units, CFU. So as you can see, Riverside Park in Piscataway is our worst site in that it exceeds the standard close to 90% of the time. And then Rutgers Boathouse and Edison Boat Basin is not far behind, exceeding the standard 75% of the time. So what that means is for all our samples, 75% of those samples exceeded the water quality standard at that site. And then in Sayreville, our Sayreville site at Cannon Park was about 50%, close to 60% of the time. Our best site is South Amboy Waterfront Park, which is actually a non-bathing beach. It's a sandy beach, and it exceeds the standards about a quarter of the time. And then Perth Amboy, Second Street Park, about 50% of the time. And that's for pathogens. That's for Enterococcus only. And then we are collecting other variables. We're also collecting precipitation data. And that's important because we want to see if there's a correlation. We're collecting these variables because we want to see if there's a correlation between pathogens and any of the other variables. So for example, you know, do the pathogen levels go up when they're right after a rain event? So we're collecting precipitation data, air and water temperature, also salinity and conductivity. Conductivity is a measure of the water's ability to actually conduct electricity. So obviously, the more fresh water in a water body, the less saline it's going to be. Ocean water is about 35. I think it's parts per million of salt. So we're also collecting pH, and so that's the acidity of the water, and dissolved oxygen. That's obviously an important indicator. The aquatic organisms living in the waterway need oxygen to breathe, just like us, and we want it. The standard is 5 milligrams per liter, so we want to see our dissolved oxygen levels above 5 milligrams per liter. We're also collecting information on the tides and also barometric pressure. So all of these variables are important for starting to be able to tease out what is possibly causing or what's correlated with high pathogen levels. Now, this is important. We're telling you this because later on, we're going to be showing you some different ways that we could potentially be displaying this data to the public and sharing this data to the public. So remember this slide.
Okay, so how does Enterococcus end up in the water, and why does the source of it matter? Okay, so so the sources actually of fecal contamination, you can see up here, the human and animal. It's warm-blooded animals that contribute enterococci into our water bodies. And the sources are these critters, these animals, warm-blooded animals, but the fecal matter ends up into our waters through broken sewer lines, combined sewers, through runoff, stormwater runoff, and animal waste. Yeah. So the issue for us is really trying to track down to which of the critters fecal matter is in the water, so that we can then figure out how to intervene. Are we dealing with livestock contributions? You have the image, the second from right on the bottom. Is it a situation where we have cows or horses pooping in our waterways? In which case, if it is, we can put regulations in place or enforce regulations that are in place for farmers and for livestock that shouldn't be going in the stream. This image is actually from the North Branch. It was provided by Joe Mish from just a few years ago. Are we dealing with human waste? So we have an image here, the second from left. This is failed sewer line, basically. We have situations in many parts of our municipalities where our sanitary sewage infrastructure is compromised. It's older, it's aging. The infrastructure bill that just passed, it will hopefully support repairs and fixes for this type of infrastructure throughout the country. We know for sure that we need it in our Lower Raritan. And of course, the sanitary sewage infrastructure, when it fails like this, that's contributing human source material. Similarly, for the combined sewer overflow, which is the image that you see on the far right, that's a CSO in Perth Amboy. So again, if we know the source, we can begin to address the problem. So as part of the monitoring work that we've done, we've taken samples to the Interstate Environmental Commission, which reports out every Friday for us in terms of the counts for fecal and enterococci. We also, Michelle did this last summer and the summer before, took an extra sample of water to the Fehrenfeld lab at Rutgers and filtered that sample down into a small sort of coffee filter type, sort of a cloth disc, where all the material was concentrated through a type of filter material. And that was then frozen for future analysis to determine the source, whether it's human, livestock, goose, etc., of the pathogens. So they were looking to match specifically for human fecal markers. So they have a panel of human. They also have, what else does Nicole have? She could have matched for goose and for horse.
Dog?
And dog, I think. But we were most interested in matching for human because we had a sense that with the CSOs and perhaps the compromised sanitary sewage infrastructure, that would be an area that we could put pressure on our municipalities to do some repairs. So what you see here in the upper right is a table of our dry day sampling in which, a small handful of dates in which we had enterococci exceeding the standards. And these were the basically the dates for which and the samples for which the Fehrenfeld lab analyzed for human fecal marker. And
Right. So this next slide here, I know this is a lot, but I will break it down for you. So as Heather said, the Fehrenfeld lab is looking for that human fecal marker that may explain some of the pollution. So what this is showing here is our sites. So you can see these little charts here. The first one being our site in Piscataway, our most upstream site. And the last one all the way on the right is our most downstream site in Perth Amboy. And this is just data from 2020 when we were doing this source track down work, this qPCR source track down work. And so the red line here indicates the standard, the water quality standard for pathogens. So anything above that line means that it exceeded the standards. The black dots are the actual samples, and the triangles are the results of the qPCR analysis, showing that they got a hit for the human fecal marker. So anything above this line here says, is showing that there was a high concentration of this human fecal marker, and that's the triangles. So you can see that they saw the fecal marker at the Piscataway site and at the New Brunswick site, and then also at the Perth Amboy site. So that's sort of generalized explanation of this chart here, that we did see that some sites we saw that the source of the fecal is potentially human.
Right. And I do want to say, right now I will unmute everyone in just a little bit, but I want to say if there are any questions, either raise your hand or feel free to enter the questions in the chat. I'm monitoring the chat right now, so. Or comments, not just questions or comments. So summer 2022, we're looking to do a lot of the same what we have done. We'll continue the sampling at the non-bathing public access beach sites, the same 6 at which we've monitored the past 3 years. We'll continue into a 4th. Both last year and so in 2021 and in 2022, we've been partnering with the EPA to basically augment, or amplify each other's water quality monitoring. In this summer, the EPA is going to basically match our sampling sites to do what is called transect sampling. So they have been doing in-channel water quality monitoring at 23 sites along the Raritan, none of which have matched up well with our monitoring sites. So what we're going to do is trying to get basically a transector cross section at our sites of basically shore proximate or riverbank proximate and in-channel samples to try and see the extent to which these samples are, the results are similar. Are we finding in-channel that there is less bacterial contamination? Is this an issue more of runoff that's getting caught in the, and with bacteria getting caught in the slower moving sort of sides of the waterway? That remains to be seen. So we'll work on that this year. So we're also planning to do some kayak, kayak trips. The image here on the right is of the outfall inventory of our watershed. This is a publicly accessible database hosted by the New Jersey Department of Environmental Protection. We are going to basically be doing some touring through to see whether these outfalls from the water are contributing actively into the Raritan, and if so, are they doing it? Is there maybe also some compromised infrastructure? Are they broken, and do they need to be fixed? So we'll do some photo documentation of that. We're also going to do a survey in English and Spanish to gauge folks' understanding of Raritan River pathogens data, of the work that we're doing to gather the data, and just to get a sense for how folks are using the Raritan and how they'd like to use the Raritan for recreation. And then, of course, we'll go sort of full loop with, taking our data to the municipalities, saying, okay, as you all saw in the last couple slides, we have an awful lot of days of exceedances. We have a pretty good sense for the human fecal contributions. We have a pretty good sense that, stormwater and rainfall is driving a lot of the exceedances that we're seeing. How can we get our municipalities to take action with green infrastructure interventions or policies or similar to minimize the impacts that we're seeing of stormwater flows on our Raritan? So here's just a graphic that kind of illustrates what I was just talking about. We are working to keep the pathogenic bacteria out of our waterway. We do the monitoring, we report out the data, we try to use that data to inform, if you look at the lower right, again, you have that broken sanitary infrastructure, sanitary sewage infrastructure. You can see the middle image, the sort of right-hand middle image with outfall at Boyd Park. And then how do we intervene? Can we intervene with rain barrels, with cisterns, underground cisterns, and rain gardens? And then we're going to see if we can gauge by measuring, after the fact of all of these green infrastructure interventions, whether they've made any difference in terms of improvements to water quality. So I'm going to show just a couple of different graphics here. As Michelle said, remember the various parameters that we sample for every week. There's the salinity, there's nitrogen, there's actually, I don't think we do nitrogen, but there's salinity and pH and oxygen. This is an example of how one county in Florida presents their ongoing sampling to the population, to the folks in the area, so that can be interpreted for recreation in freshwater waterways. So we call this a dashboard visual summary of the water quality data parameters. And it gives you a sense through this little meter here as to whether the particular parameter is meeting or exceeding the thresholds.
So take a look at these and see which ones you like, which ones speak to you.
Right.
And if you had your way, how would you want the data presented to you? These are some options. We've stolen some of our favorites to show you, and we'll hopefully have the opportunity to create our own communication tool,
Right.
online communication tool of this data.
Right. So here's the dashboard approach. Here's what Pinellas County, Florida calls an atlas of, a map-based atlas that is searchable, that basically communicates whether there are high bacteria levels, other pollution reports, algal blooms, red tide. So you can see on the right-hand table, there are color-coded symbols, either triangles or little, they look like, conversation bubbles. And if we were to click on this link for Pinellas County's Water Atlas and go there, you would be able to open up each one of these bubbles to get more information about the site and the types of pollution there. And then there's basically a model, so we had, first the dashboard summary and atlas summary, and now we have a water quality model that takes both actual and predictive information and estimates risks for bacteria levels throughout the year. Gives folks sort of a big picture sense for potential risk in the waterways, in this case, for combined sewer overflows. So we'd have to imagine that this is the risk that probably overlaps quite well with the annual precipitation cycles. So poll question 5 here. Let me open up the poll. Hold on one second here. Which summary approach do you prefer as an overview visual of water quality data? Is it open for you all?
Yeah.
Yeah, great. Okay. So the water quality dashboard, the water quality atlas, or the water quality model that combines the actual and predictive information. Okay. Okay, I think that's, there we go. Okay. Great. Great. I'm going to close the poll. I think everybody's answered. Share out the poll results. Michelle, can you see them?
Yeah, I can. And it looks like 71% of folks said that B, the water quality atlas was their favorite.
Great. Okay, so that's with the pinpoint location on the map, zeroing in with on specific points that also I should have mentioned allows you to open up the data and you can download that data. Okay, great. Okay, so Michelle, do you want to talk about going forward?
Sure. Yeah, so we already have what's called the Raritan River Hydrologic Observatory. There's the website for it there, and we are working with the Rutgers Office of Research Analytics to kind of rethink this so it can be a better data communication tool. So I don't know that initially it will be as spectacular as some of those others that we saw, but we're working long term to get it there. And be able to have the data that we are collecting made available to the public through an online map like this, an online software. So that's sort of a 2022 goal for us. And so we want to know from you, would you be interested in accessing the data that we're collecting so you can perform your own analysis? Maybe through teaching, maybe through demonstration, all sorts of opportunities. So that's the next poll question.
Right. So would you like to be able to access our Raritan River water quality data so that you can perform your own statistical and other analyses? Okay, great. And I want to say too that what we're planning on doing and what we have been doing with the Rutgers Office of Analytics is developing models so that we can drill down even further to what some of the contributing factors to our high levels of bacteria may be. So it may be a case that, so one of the things that we want to do is not just provide information about when there's risk, after a rainfall, but also perhaps present information about how long it takes for that risk to sort of dissipate. And with the data that we've been gathering, and with input for a variety of other parameters, we're starting to get a better sense for the role that, for example, tidal flows might play in influencing the increased or decreased dissipation over time for some of the numbers that we're seeing for the high pathogens concentrations at certain locations. Okay, so
Did folks answer that question?
yes. Oh, didn't the poll question pop up? Sorry.
It did. So I think you just have to close it.
Sorry. Poll results, that's what I need to do. I didn't. What are the poll results here?
Yeah, so most everybody said yes, that they
Yeah,
would like access.
not surprising. Sorry. Okay, great. With one no answer. Okay, so of course, there are many ways that you can get involved. We have the ongoing water quality monitoring, and we have trainings coming up for that May 19th, an in-person training, 5 to 8 at the Middlesex County Earth Center. There's the URL here on the slide, and I'll put that in the chat box as well. And then also in the chat box, I'm going to put this right now, is, our survey. So you all were guinea pigs for portions of our survey, for a couple of the survey questions, in what we hope will be a very well responded to questionnaire. We want to get a sense for how folks are using the data that we are gathering, because the more folks who are paying attention to what's going on in the Raritan, the more attention and support we can rally for cleanups of the waterway, for policy changes in the upstream areas, for green infrastructure interventions, for the rain gardens, for the detention basins. So that survey is going to help not only with outreach around the pathogens piece of things, but also help us do the advocacy work around the changes that need to be made upstream. So anything else, Michelle, on this?
That's the QR code there. You can scan it with your phone if you'd like, and then we'll be posting this out, the survey out on social media. We really do need people's help with sharing it and getting it out there in your Raritan communities. So please, when you see that come out, please make an effort to share that with folks.
Right. So that is pretty much it for us. I have tried to unmute everyone, so that we could have a little bit of a discussion here. Get your feedback, hear your voices, and get a sense for your interest in the waterway, suggestions that you may have for us going forward with this work. Perhaps a comment about the work that you yourselves are doing to improve the health of the Raritan or to better understand the health of the Raritan. So please feel free to unmute yourself, and happy to chat. Are folks able to chat and unmute themselves?
Yeah, there's just a few people who have microphone capability.
Okay.
The others seem to be either they're muted themselves or
Okay.
don't have the ability to unmute. You can
Okay.
also put any questions that you may have in the chat box.
Heather, can you hear me?
Yeah.
This is Ken Yetman.
Hi, Ken. How are you?
Okay. You're using the pathogens, the E. coli and the coliform and the bacteria, but it is not bacteria. Contamination is primarily a problem when it's from a human source and is a health problem, health issue. And from when the data you showed, it looked like the biggest problem was around New Brunswick boathouse station. Using it in other areas, you're going to get some bacterial runoff, from mostly dogs and cat waste in urban areas, but it is not a human health issue. And I'm just wondering how much you've done to identify the sewage sources, especially in New Brunswick.
Yeah, so we're seeing pretty consistent high Enterococcus levels in Piscataway and New Brunswick and Edison. And you're right, those are obviously incredibly urban areas. And we have been doing source track down work to look to see, well, what, as you said, what is potentially, or any of the are we seeing high Enterococcus levels? Can it be attributed to human sources? Are there leaks in the sewer lines? So what we have seen, the data so far that we've seen is that in Piscataway and the New Brunswick area, the answer is yes. Some of that may be human. So you're right, that is definitely a human health concern. Also down in Perth Amboy is, we sample right at the combined sewer outfall in Perth Amboy, and we did get a hit there also for the human marker. and that's, you know, probably attributed to that,
Yes.
the CSO.
Yeah. But I mean, you can get high bacteria levels associated with marsh systems and, just natural types of sources, which, so I mean, when I've looked at data like this in the past, I've always kind of honed in on the human source because people
Right.
kind of react to raw sewage in their water.
Yeah.
Uh, as you don't want excessive coliforms from any source, but human source tends to be that's the one where that's the one that makes people sick.
Right, right. Absolutely. And so we've done some of that work already to look to see if it is human. And then we're also we just had a great meeting with the DP, and we're hoping to do more of that with them to try to figure out, you know, what is the cause of this.
Okay.
And that we have funding, or we are anticipating, we have been approved for a 3-year Sea Grant Consortium study to continue with the qPCR for human fecal contribution. So we expect for the next 3 years to continue to, for each of these sites, drill down to get a better sense for whether it is human fecal.
Yeah, I'm not familiar with qPCR. I've dealt with genetic testings, which we collected isolates from different sources and then determined how much was human, how much was dog, cat, goose, things like that.
Right. So with the qPCR, generally what's done is, you do have the isolate, and there are different panels for different regions. And so we've been working with a panel that was developed originally for the Navasink, which is quite comparable to the Raritan. And now we're also developing a panel specific for the Raritan as part of the Sea Grant. Yeah. But again, the image that is up on the screen right now is really showing where you have the source tracking with human fecal marker on the triangles here. So we have it for Piscataway, we have it for New Brunswick, we have it for Perth Amboy for this particular set of samples. EPA has also found some, and they, as we mentioned, they have been sampling at 23 additional sites in 2021, and will continue that in 2022. And they have also been identifying human fecal contributions.
Okay.
Did that answer your question, Ken?
Yeah, it's I just in the past have kind of ignored fecal, bacterial levels from non-human sources and kind of just concentrated on the where it's a health issue.
Right, and that is what we have been focusing on primarily. I mean, certainly for as urban a waterway as the Lower Raritan is.
Yeah, but I mean, for example, in Edison, on the last one on the 2020-022. I guess that's 1022. It exceeded the level, but it was not human, and so in your thing, you would have given it a red smiley face or a red frowny face, but yet it wasn't it's much less of an issue than it was in New Brunswick, the same time.
Right. And that's so what we're trying to do is provide as immediate set of results as possible
Yeah,
for
I understand the
for
limitations of.
decision making.
Yeah.
And hopefully we'll have tools and resources that have more rapid response. That's one of the approaches that we're adopting through the C grant. Grant is a faster assessment of genetic contribution for so that's something that we're trying out for the next 3 years.
Okay.
This is also the same methodology that's used at the bathing beaches on the shore. They're taking these exact same. They're sampling for Enterococcus at the bathing beaches, and then they will drill down further if they see that there's exceedances there. So we're just trying to collect the same data that they are collecting there using the same methodology. Other questions from anyone else or feedback, advice? All right.
So I don't see any in the chat. But a huge thanks to everybody for joining us and for caring about the Lower Raritan. So I know all of you are advocates and activists for the waterway in one way or another. So thanks for that as well. We did share the survey in the chat. We hope that you take the full survey and please share it with friends and family. We're trying to, as I said, get a good number of folks participating in that so that we can demonstrate an interest in the waterway and maybe bring some more money to the Raritan to clean it up. Anything else, Michelle?
No, that is it. Thank you, everybody, for spending your Thursday evening with us.
Yeah. Thank you. Good night.
Thank you.
Good night.